इसमें हाल में इतने लोग हो गए, मुझे लगता है इसको सीधी कम रखिए। लाइट बस चलेगी।
Last one. Which one, sir? Thanks. I'll copy the whole thing if something happens, sir.
Can we start? Good afternoon, everyone. Good afternoon, everyone, and I would like to welcome you all to the third session of the third day of INCD 2022. The session is on inflammatory diseases, and it is going to be consisted of one keynote lecture and 5 invited lectures. I would take this opportunity to welcome the chairs of the session, Professor Vinod Kumar and Professor Shivnath Majumdar. Standing here and welcoming them, introducing them gives me a mixed feeling. I feel very privileged to introduce my teachers as chairperson. I am also falling short of words, and I'm sure the 1 minute that I have cannot be used to summarize their academic career, the prolific academic career in but I'll still try. So, Professor Vinod Kumar has had a prolific academic career which does not need any mention. Sir did his PhD in 1981 from Banaras Hindu University and then went on to give his academic contribution to several institutions and universities. To name a few, he has been the visiting fellow of Max Planck, Germany. He has been the visiting professor, Department of Biology, Texas University. Sir has been a recipient of several prestigious awards. Very early on in his career, he received the Young Scientist Award from the Indian Science Congress Association in 1986. He was the Young Investigator Award from the Gordon Research Conference in Pioneer Cell Biology in 2000. He received the P. Govindarajan Gold Medal, Society for Reproductive Biology and Comparative Endocrinology in 2006. Vijay Usha Shore Scientific Research Prize, University of Lucknow in 2008, the Platinum Jubilee Lecture Award from Indian Science Congress Association 2010. Very recently, Sir has been the recipient of the prestigious Ascof's Royal Prize in 2022 and currently he is the UGC BSR Fellow in the Department of Zoology, University of Delhi. I welcome you, Sir. Our next chair is Professor Shivnath Majumdar, currently Professor, Dean and Proctor, Faculty of Life Science and Biotechnology, South Asian University. Sir did his PhD in 1997 from the Postgraduate Institute of Medical Education and Research, Chandigarh. Sir has been the recipient of INSA Teachers Award. He is the member of several prestigious societies, Indian Immunology Society, Indian Society of Cell Biology, Indian Society for Molecules, American Immunology Forum India, to name a few. He has given his academic contributions to Vishwabharati, to Banastali Vidyapeeth, as well as to our very own Department of Zoology, University of Delhi. Sir has mentored several students and has several research articles to his credit. I welcome you, sir. And with this, I would like both of you to take on to the session. Thank you.
Thank you, Dr. Mamta Tripathi. Really very embarrassing note. It's all in Google, and I am retired now, so fused bulb. So that doesn't matter. But I'm really privileged to co-chair this session with my senior colleague, Professor Shivnath Majumdar, and I proudly hand over this mic to him because he is the right person for this particular session. So here on Professor Shivnath Majumdar will take care, and I'll try to listen, catch up some of the immunological. Thank you so much.
Also, I'd like to thank the organizers for giving me a chance to participate in this session. And I have a very nice time together. Our first speaker is Professor Andrew Srivastava. And it's very difficult for me to introduce Andrew Srivastava because although I knew we were friends, we used to fight together, we used to do everything together. We were wonderful. So, this is now the Professor of Zoology, and you have everything over here to read it out.
So do I have the permission?
Yes, today at least you are asking me for permission.
Thank you. And, thank you, everyone, and good afternoon. And it's really, I'm very happy, in fact, that I'm getting the opportunity to speak in front of my teachers. There are two, three teachers who I have actually learned a lot. Professor Bharat Agarwal, Professor Madan Mohan Chaturvedi, and in fact, coming in research is because of Professor Madan Mohan Chaturvedi, because the interest in science actually was inculcated by him, and not because he's sitting in front and saying that is the truth, sir. I've always been inspired by your way of teaching and the way you have inspired the students, right? When we were also students, and all the students here would acknowledge. And as far as getting into the field in which I'm now working is the contribution of Professor Bharat Agarwal, because I had the opportunity, good fortune to be in his lab for a couple of years. And that is when I got interested in immunology and inflammation. And that is from there only I actually picked up this area. Now, the work which I'm going to present today is a newfound interest. Earlier, I used to work with macrophage. My PhD supervisor, Professor Ajit Sodhi, and I shouldn't forget to actually mention him and to actually pay my respect to him, because he is another person who has been very, very instrumental in where I am at this point in time. So the newfound interest, which I say is neutrophil. Up till now, we have been paying lots of attention to macrophage, and macrophage particularly because they have the tendency to infiltrate in the areas where inflammation actually is. Whatever kind of inflammation, we will find macrophages sitting there. So, and especially in tumor, there is infiltration of these macrophages, and I was very much interested, and I used to work, and I'm still working with macrophages. But during the course of studying macrophages, there were few observations which actually attracted me towards neutrophil. And when I started looking, and this is where I would say that these are some of the limitations of the way we work. When we work, we become so focused that we do not look beyond what we are working. So I used to get focused, my lab used to be focused only on macrophages, and we were not looking on the other cells. But during the course of my journey, I got to see that when the cancer actually develops, there is tremendous change in the number of neutrophils, which we actually found out not because we were looking for neutrophil, but accidentally. So here, and then when we started looking into the literature and other things, what we and during our research also, what we find that when we say inflammation, what we are actually talking about the fire within, and as long as this fire is in control, it is good, it helps us. But the moment it becomes uncontrolled, it can lead to several problems. And so this is what it is. It says as long as it is acute, it helps us. The moment it becomes persistent and chronic, it can actually result into a number of different kinds of diseases. And the other key to inflammation, all of us know, that is neutrophil. And when you look into inflammation, you will find right from head to toe, any kind of inflammation, any organ, you will find inflammation is resulting into various kinds of diseases impacting various organs. So inflammation, as long as it is acute, it can actually bring about a beneficial effect. Once it becomes chronic, it starts causing problem, and then later on it can actually become systemic chronic inflammation, and that might actually result into various kinds of issues. It may have a systemic effect, and as you can see in this case, as you can see, if we talk about take the example of visceral adipose tissue inflammation, this inflammation may cross talk through the adipokines and other effector molecules to the brain, and through the brain various organs are getting impacted, and these organs in turn may cross talk with the inflamed tissue, and it might actually result into various spread of the inflammatory conditions. Various organs may get impacted, and the major role played may be by the phagocytes, it may be macrophages, or it can be neutrophils as well. And as this cartoon shows, that there could be number of mediators which are released, but this crosstalk is majorly mediated by the neutrophil, and a very beautiful review article was published recently in 2020, which actually shows what are the various mediators, and especially the neutrophils play a very important role in the crosstalk. So, what impacts, what are the causes, and all of us by now, through the course of this whole discussion, have come to now realize, and we already know that be it xenobiotics, disturbed sleep, chronic stress, diet, dysbiosis, obesity, physical inactivity, or chronic infection, you name, all these lifestyle-related things may actually impact these cells, and later on it might actually impact all our organs. So inflammation does have a systemic effect. But, and the key are myeloid cells which originate in the bone marrow, and from bone marrow various cells are originating, but today we will confine our discussion to the neutrophil. Now, as the beauty of the cells of the immune system is that they show tremendous plasticity. And this plasticity basically means, what I mean here is phenotypic plasticity. That means depending upon the area where they are living, or where they are going, or from where it is coming, it may show different phenotype. So so like any other immune cells, even the neutrophils are found to show various phenotypes, say neutrophil, or you say T cell suppressive neutrophil, or neutrophil which may show enhanced mitosis. So, and all these different phenotypes can be identified depending upon various surface markers which they express. And now people have started realizing the importance of these neutrophil in various disease conditions. All of us know they originate in bone marrow. Through successive course of development, they actually develop into neutrophil. There are around 50 to 70% of the cell population are the neutrophils. And what is very interesting is only 1 to 2% of the mature neutrophil come into circulation. And they remain in the circulation whenever there is requirement, whenever there is a stress condition, there is a call, then only these neutrophils infiltrate into the tumors, into the inflamed tissue, and may bring about resolution of the problem. They may function while the important point of neutrophil is that they have lots of granules or the cargoes which can bring about their effector function. They may function via degranulation through phagocytosis, and newfound technique is that they may show mitosis, wherein they can send their cargo specifically through mitosis to trap the various pathogen and resolve the problem. So neutrophil plays an important role both in health and diseases. And when at the site of infection, once the neutrophil function is over, it should be cleared, and so as to maintain the tissue homeostasis. But as long as they remain there for prolonged time, they accumulate, they may cause actually tissue damage, and that can actually initiate chronic inflammatory state. So, and people have found that if there is elevated or increased recruitment of neutrophil or heightened function of these neutrophil, excessive release of the granules which they actually have, you will find that they are being implicated in multiple diseases involving various organs. So this is just an example. If we talk about respiratory disease, neutrophil have been recently in COVID-19, it has been implicated very severely. And then you can see, they function via the release of the cargoes, the reactive oxygen intermediates, proteases, and other things. So now the question is whether neutrophil plays any role in cancer. And why this question was that until very recently it was said that they are neutral. They have no role to play as far as cancer is concerned. But recently people started paying attention, and the major attention was paid into tumor-infiltrating neutrophils. So the ones which are infiltrating, they are activated, and they may actually cause certain kinds of problem. Okay, but not much was actually known, and not much has been worked on the systemic effect of neutrophil. And we actually wanted to study that whether these neutrophils which are there in an individual, especially we use mouse model, whether the neutrophil which are found there, do they have impact on the tumor macro environment? And we wanted to actually study that. The question was whether and how neutrophil contribute in mediating any kind of systemic effect in the cancer. All right. So we tried to look into the numbers and whether they are infiltrating into the into various organs or not, and if they are infiltrating, what is its function, what kind of phenotypic variation they are showing. So in order to assess its function, we actually utilize a mouse model wherein we actually studied the neutrophil on various time point of cancer growth, that was on day 0, starting point 7 and 14, so as to see with progression what changes are actually happening. And this whole study had started with this observation. The story which I said, it came later to this part. Why? Because one of my PhD students, when she started, we actually started looking for macrophages, but when I asked her to look into the blood profile, what she found that when you take a look into the blood profile of a tumor growing mice, you are seeing that in the tumor, in the blood, there is heightened number of very high increase in the neutrophil count. And as the morphology shows, the neutrophil number was very high. And then we looked into the TLC, and then we also did the DLC, and we find that the polymorph numbers are actually, or the neutrophil numbers are actually increasing gradually with the increase in the, with the progression of the cancer. On the contrary, the lymphocyte number was actually going down. And when we looked into the literature, we found that this polymorph to lymphocyte ratio actually is a predictive marker to say whether in cases of growing cancer. Just one, give me 1 second. So then whether or not it is a neutrophil, we actually checked for the marker, and Ly6G was used. And as you can see, with progression 7, 14, and 21 days, it is increasing in number. We did FACS analysis that also showed that the neutrophil numbers are increasing in the blood. Then the next thing which we could have done is to see the enzyme function and of the various, such as liver function, kidney function, and what we saw that with progression, the enzymes are also showing variation, especially the ALT, the amount is getting high. Similarly, the urea is showing increase, albumin is showing increase. So there were indications that the organs are actually getting. This was the preliminary data. Based on that, we actually did the whole story. Then the question was that do we see these cells or any kind of, if your organs are showing different function, there is increase in the enzyme level or there is difference, we looked into the histology of it, and what we found to our surprise that in all these organs, lungs, liver, or say for example spleen, or you talk about peritoneum where we had actually injected the tumor, in all the cases we are seeing there is increase in filtration of these polymorphs. Now the question was that whether these polymorphs are neutrophil. Similarly, we did the Ly6G staining for these, and we found there was tremendous increase. If you see here, look at the peritoneum, look at the lung tissue. If you look into the kidney tissue, again, you can see that there is very heightened increase of neutrophil infiltration. There was significant increase in the neutrophil infiltration. So in this first observation, the preliminary data which we were actually, which we saw, the observation was that there was increased count of neutrophil in the peripheral blood. There was tremendous increase in infiltration of neutrophil in the vital organs. We were not looking into the tumor infiltration, we were looking into the organs, the peripheral organs, the places other than the tumor. There was disturbed histoarchitecture and altered biochemical enzymes. So the question was to examine whether systemic impact of neutrophil, what is because of the, is it because of the systemic impact of the neutrophil? And then the most obvious thing was to find out what is its functional change. Is it showing heightened activation, or is it contributing because of the neutrophil function? And as all of us know, neutrophil actually have activation. They have acidophilic granules. You have another granules such as collagenase or gelatinase, or there are a number of enzymes which are actually there present in these granules. And upon activation, they may work via granulation, phagocytosis, or netosis. So, in order to bring about the killing. So, this is how it works. So, again, in the same setup, we try to look different enzymes so as to understand the tissue change which is happening. Is it because of the release of these enzymes in these tissues? So, the neutrophil elastase is the most abundant enzyme present in the acidophilic granules, and these hydrolyzes the elastin and other extracellular matrix components such as cadherins, fibronectins, and all. So we wanted to look into that. And if you look into this, if you look into the gradual progression, you can see how much increase is there of neutrophil elastase. Similarly, we looked for MPO, another important enzyme, and you can see exactly similar trend that there is increase in the MPO with progression of cancer. And if we look into this is the nucleus, or this is the blood cells of the neutrophil obtained at different time points. And the most remarkable thing which you can see here, that the nucleus is actually getting highly fragmented, and this is the hallmark of the change in the phenotype of the neutrophil in cases where the cancer actually is progressing. So even like macrophages, the neutrophils are actually N1 or N2 type, which is one is known as tumor promoting, the other one is known as tumor supporting type. And if you look here, if we actually looked into the Ly6G staining is for confirming neutrophil, MPO and NE staining, and when you look into this, you can see these neutrophils are expressing, the ones which were present in the peripheral blood are also expressing these enzymes in much higher quantity as compared, and the phenotype is also getting changed. So, if the infiltration contributes to high elastase with progression, all right. So in the blood, in the ascites where the tumor is growing, as well as in various organs, liver, lungs, spleen, and peritoneum, we also looked at the transcriptional level. And as you can see, DL actually says it is the Dalton's lymphoma which we have utilized, and you can see how much increase in the expression of messenger RNA level is there in the cases of tumor. Now, similarly, if you look into the immunohistochemical staining of the tissues, you can see the same situation in the liver, in the peritoneum. The upper panel is for control, and the lower panel is for the lower panel is for the tumors. And if I go beyond spleen and lungs, you can see in the tumor condition how much increase in the enzyme levels are. Similarly, MPO, you can see at the transcriptional level, there is increase in the expression of MPO, and you can see through immunohistochemical staining as well that there is increase in MPO expression in all these tissues. So, and then this is another very interesting point, the ROS level in these organs, liver, peritoneum, spleen, and lungs. You can see how much increase in the ROS level is actually seen in the cases with increase in tumor enlargement. Then comes whether the collagenases and gelatinases are also showing any change. The gene expression levels can also shows the similar trend. Whether it is MMP 8 or 9, it is showing increase. We did the gelatinase assay. in order to confirm the activity, there is increase in the activity in the tumor tissue. So with this, we were very convinced that the neutrophil infiltration contributes to high elastase release in the vital organs, MPO release, as well as there is tremendous increase in the ROS levels, and there is increased activity of MMP 8 and 9, which actually suggests that the changes in the organ function probably is because of the infiltrating neutrophil, and it might be contributing to the damage. And this part of the story very recently got published into the immunology letters. Now the question is, and this is a very basic cellular study, no molecular study I have done here, no signaling aspect. This is just to see how exactly the neutrophil may be playing a role in actually having a systemic effect. So this, we were kind of convinced. Now the question was whether this whole, because these enzymes are also produced by the macrophages. So what is the contribution of macrophage? We are taking these enzymes in the enzymes. So is it being contributed by the macrophages or by the neutrophil? But what we are seeing, and we think and we assume it needs to be actually ascertained, but what we see, because there is a correlation, there is very heightened increase in the neutrophil count, so probably these are the ones which might be contributing, but this is worthwhile to find out that whether it is being contributed by macrophages or not. Next, we wanted to see whether this can be actually tamed. These are hyperactive neutrophil, and can it be tamed? And can any immunomodulator actually bring about some sort of calming down of these neutrophil, and can it be useful in actually the in survival, or maybe it may be helpful? So we actually utilized Tinospora cardifolia, and this we used a crude extract, methanolic extract. We did not do anything except for we use the extract. And in the same setup, we actually use this extract at different time points. We injected the extract right at the time when tumor was being transplanted, so as to see its preventive effect. And in the second set of the group, we utilized, or they gave the mice after 7 days of tumor growth, and then later on after 14 days of tumor growth. So just to see from what to what level the Tinospora we are using may actually have an effect. So if you see this graphs, one has to pay attention is to the way that this 0 represents simultaneous tumor transplantation and TCE administration. This is after 7 days of tumor transplantation, and this is after 14 days of tumor transplantation. And these are tumor mice. All the parameters of the tumor mice is represented by DL, right? And then when you see, if you look at the 0 time, when you are in any parameters, you will see that these parameters, when it is given along with the tumor cell, when the tumor was being transplanted, Tinospora was found to be highly effective in preventing the tumor growth. We will see one by one the various parameters. So this is the TLC count, the total leukocyte count. These are the polymorphs, and these are the lymphocytes which was found to be significantly reduced, was much better when it was actually, so the blood profile was seen to be actually reclaimed or restored, right? Similarly, if you look into this is the DL condition or the tumor condition. If you give simultaneously, it is more or less like the control. After 7 days of tumor, when the tumor has grown slightly, still it has an effect, but after 14 days, you don't see that kind of an effect. Now, in this case, this is the 0 day, 7 day, and 14 day. Look at the 0 day, almost like like the control. It showed tremendous effect on in preventing, and at early stage also it was found to be fairly effective. If you look into the ascites and the serum for the elastase, again you can see how significantly there is prevention, and even at the early stage it is significantly showing restoration of or reduced release of elastase in the lungs, liver, spleen, and peritoneum. Everywhere we are seeing that there is significant protection as far as Tinospora administration was concerned. Which it is again beautifully shown when we looked into various tissues. You can see at the 0, 7-day, and 14-day condition. Right? These are different, we also did ELISA in different ways, we checked into it. MPO, and this is the most interesting one. If you see the DL condition, the reactive oxygen state, and look at this at zero time administration when it is given simultaneously, how significantly actually it is blocking the ROS production. Even at after 7 days of tumor transplantation, the effect on the ROS production is very much actually reduced. So, and then when you look into the MMPs, it regulates the MMP expression in various organs, be it liver, be it lungs, be it spleen, or peritoneum. In all the cases, the MMPs are expression is again showing the similar trend. So TC restores the tissue histoarchitecture. This is the normal tissue, and if you are giving it at the 0 simultaneously, it is able to actually prevent the lungs histoarchitecture, the liver histoarchitecture. Even at 7-day treatment, you can see the effect is pretty significant. This is the normal peritoneum. You see here at 0 time points, you can see in the spleen as well, the normal architecture is very much intact. Similarly, you can see the serum enzyme level. If this is important, the AST almost like normal, urea almost like the normal condition. So the point which I wanted to emphasize that the TCE, when it is given, and the dose which we actually tried, was able to prevent the tumor progression or tumor growth when it was given together, and when it was given even at the early stage, it had significant impact on the survival of the animal. So if you can, the 0-day, 7-day, and 14-day, you can see normally this mice grows somewhere around 24 to 25 days, but the moment you give at 0-day, it is surviving almost like 80 days. After 7 days, if you give, it is somewhere more than 60 days, and even after 14 days, there is increase in the survival. So maybe that how it can be given, but one point which is very, very important to understand, or what we realize, that if we actually have some immunomodulators which can actually have the ability to tame its activity, neutrophil function, it might be actually useful. This is a very preliminary small study just to say that if there are certain immunomodulators which can actually have an impact on neutrophil function, if they are calmed down, they are tamed, then it can have an impact. So Tinospora cardifolia in our preliminary study actually shows that it regulates the neutrophil number in the blood, as well as infiltration in the vital organs. It regulates its hyperactivation. It can, and if it is regulated, the activation is regulated, it may restore the tissue histoarchitecture and will limit the growth or the increase the systemic deterioration may be actually controlled, and that might actually add to the survival, better life which a tumor patient actually faces major problem actually happens not because of the tumor, but because of the deteriorating systemic condition. So if that also can be controlled, then it might be of use for a better survival. So neutrophil count, so taken together, we strongly support the hypothesis that neutrophil might be majorly responsible for the systemic manifestation observed in the cancer patient, which we actually come from the first part of the study, wherein we are seeing the systemic damage is majorly contributed by the neutrophil, and taming these hyperactive neutrophil might have certain preventive and maybe with combination, it might also help in therapeutic approach and may increase the survival of the person of patient and may actually help in better life quality. What could be the future direction? Earlier, it was known that there are pro-tumoral role of neutrophil. The number increases in blood tumor site. They are involved in various parameters of the tumor growth in the microenvironment. Our observations suggest that it has a systemic role to play. The systemic damage during cancer growth is probably because of this heightened neutrophil, and what we need to do is to look into how exactly it can be tamed. One such cautious or thing which we have tried is to look into immunomodulator. Can it actually tame it down? And there could be certain others. We need to also look into the fact that from where these neutrophils are coming. Are these neutrophils, just 1 minute, are these neutrophils coming from the bone marrow or they're coming through the tumor? What is the source? What is the root? Where exactly and when is it getting activated? Those are the questions which needs to be actually addressed. But one important point is that they do play a very, very important role. So what I always say that what is the most important thing which one needs to do is to stimulate the phagocyte, be it macrophage or neutrophil. These are the only genuine things. Drugs are usually dilution, and this is my team. Our lab actually works in immunomodulation and tumor development. Major work is on macrophages. This is the girl Kavita who recently got her PhD on this part of the from this part of the study, and others are working on macrophages and other things. There is another area where we are actually diversifying is to look into neurodegenerative diseases, wherein I'm collaborating with one of my colleague in the department, Professor Namita Agarwal, and we are looking into how macrophages play an important role in neurodegeneration using the fruit fly model. These are my students, these are collaborators. Acknowledgement for the microscopy facility and the funding, and that's how I pay my gratitude and acknowledge and thank you for listening to me.
So we have 5 minutes for any questions. Yes, yes, sure.
It's very interesting. Is it possible because they increase the number of neutrophils? But if you take individual neutrophils, then the differential expressions of, you have shown the differentiation of neutrophils. Which stage is the one which I think we study that which stage will be more crucial or just simply number per se?
So, sir, in generally, that is what I don't understand, but what is very important that in generally when there is inflammation, especially acute inflammation, when the neutrophil, it is the tendency of the neutrophil is to reach to the inflamed site. So these neutrophils, when they reach to the inflamed site, they get activated by the milieu, the environment there. But the moment that they perform their function, they actually undergo apoptosis. They have a very short half-life after activation. But what we have seen in the cases of tumor growth, that these neutrophils, instead of undergoing apoptosis, they are actually surviving and they are remaining in the system for a much longer duration. They don't show apoptosis. So there is not that kind of a case. Now the question is that is it that more granulopoiesis is happening, and the moment there is more granulopoiesis, more and more neutrophil is being released because there is a stress condition whereby the tumor is going. Now, are they activated right at that point and reaching to the, because they are infiltrating, they might go into the various organs, and so, or they are going through the tumor. That part of the story I do not know. Maybe we need to actually look into that. I hope I answered you.
You have a question? So it was, the first question was between
So,
See, in this case, we have injected it, right? But one can also, I also tried giving orally. There, the dose was not actually getting adjusted. It was not constant. Kabhi zyada ho raha tha, kabhi kam ho raha tha. So it was not giving me. But here in this case, I have injected it.
So,
I mean, for me, it's just kind of learning. What I couldn't understand that you had, put the tumor and looked into neutrophil, right? Is it the neutrophil gets haywire and the tumor develops? Or the tumor has developed and it has systemic effect through neutrophils?
Yes.
So what is your hypothesis?
See, the thing is, what I am hypothesizing at this point in time is that the tumor grows and the neutrophils, because of the stress condition, neutrophils are getting hyperactivated. And from tumor, there are a number of mediators, because tumor is actually infiltrated by immune cells, be it macrophages, neutrophil, T cells, and other cells.
So this time is not correct. But then it can be happening by any stressor.
It can happen by any. So that is
then if that's true, then it's not cancer specific.
Quite possible. That needs to be looked. But then the kind of systemic impact in growing cancer condition is much higher.
Bigger enemy is there, damage would be more.
Yes, yes, I agree.
I mean, is there a way that you can chamber the neutrophil and then your rat or mice, whatever, becomes more susceptible to A versus B disease?
Yes.
That
can be looked at.
Yes, that can be. Yes, ma'am.
Nice talking. Just curious, what happens when you administer Neosporin in terms of neutrophil infiltration? Does it get?
It goes down.
It goes down.
It goes down. But it goes down.
any other question? The speaker is absent.
So our next speaker is
Sir. I would now request Professor Vinod Kumar to felicitate Professor Vishnu Srivastava for delivering such an insightful lecture.
So we move on to the next speaker.
He's from the Department of Biochemistry, Elements and Material Sciences, Guwahati, Assam. 20 minutes plus 5.
10 minutes extra looks like, sir. Thank you so much.
It's okay, that's what.
Okay, thank
We
you.
received this signal from the organizers.
Okay, thank you so much, sir.
So I will move from here. You will change the slide from here.
You will change?
Okay, I got to change, sir. But if you want.
Okay, I will change. I can use this one also, right?
for changing, sir. For laser
Okay.
user trackpads.
Okay. So thank you so much, Chairperson Professor Majumdar, for inviting me here. And at the outset, I would like to just give a comment about Professor Vinod Kumar, what he said in the beginning, that it is a fuse bulb. I would like to say, sir, it is not a fuse bulb. You, the retired person like you, are a great inspiration for us, for this generation. And second, I would like to mention about many things. Professor Anju Srivastava has made my job easy. She has already expressed many of my feelings about Professor Bharat Agarwal and Professor M.M. Chaturvedi, and I'm glad that Professor Chaturvedi is also sitting here. Third, coming to my topic, as you can see, I'm going to talk about how we can bridge the gap between traditional medicine and modern medicine, and through modern biology. And this aspect will be very brief. What I'm going to talk about, I'm going to talk about something about modern medicine. Then I'm going to talk about traditional medicine, and then how we can bridge, and then I'm going to talk about some molecules that I have been working in my laboratory. So having said that, so I will just take brief introduction. This is where I come from. As of now, I am at All India Institute of Medical Sciences, Guwahati. I am heading the biochemistry department. I am also looking after the research section, and this is one of the fast growing aims in India. You will be aware that in India there are now 22 aims, and we are now taking the third batch of MBBS students, and we are also going to start MD PhD program this year only. And before moving to this university, this institute, I was with Banaras Hindu University, with which I have a deep respect. I am still affiliated with the Banaras Hindu University as Anlian. So, and you know, I am from biochemistry department. And this is, if you talk about the modern medicine, you all will be aware, these modern medicines are also called allopathy medicine, and these are also called alternative medicine because they have been established as an alternative to the traditional medicine. These are scientifically proven and well developed after practicing on real patients, and most modern medicine that are available for cancer today, they produce several side effects, and they are unaffordable to greater than 80% of the world population. This is well-known fact. Patients on modern medicine develop cancer-associated symptoms such as depression, neuropathy, pain, loss of appetite, lack of sleep, and fatigue. These are cancer-associated symptoms. You know, when the patient develops the cancer, they die not because of the cancer, they die because of the cancer-associated several symptoms. And cancer patients develop another aspect of the modern medicine is that the cancer patients develop resistance to the medicine over the course of treatment time. And you will be also aware that it takes around 12 to 15 years and almost $1.5 billion for any drug to make to the market. And most modern medicine are basically rationally designed based on mono-targeted, most specifically specific targets. So if you talk about the potential of the traditional medicine, these medicines were basically originally believed that these are developed based on the theories, belief, and experiences that are indigenous to the different cultures. And however, in advances in technology over the year has provided the scientific basis for the value of these traditional medicine. And these traditional medicines are used in the maintenance of health as well as in the diagnosis, prevention, and treatment of human diseases. And these have been used for many generations, and therefore these agents have well-tested safety. And so approximately 80%, you will be aware that approximately 80% of the population in Africa and Asia still use traditional medicine for primary health care. And traditional medicine, it is very well known fact, these are designed multi-targeted naturally, and these can be used either alone or as an adjunct to current chemotherapeutic agents to enhance the efficacy and minimize the chemotherapy-induced toxicity. And as I discussed, most modern medicine, they are basically designed against some specific target, and these are basically we all know that many chronic diseases, including cancer, is basically caused by dysregulation of multiple genes. And therefore, now there is change in the paradigm. That is, we are shifting from mono-targeted to the multi-targeted agents. So this raises a question whether the process of drug development is moving in the forward direction or in the back direction. And because we are moving towards the multi-targeted agents, therefore this basically is that indicate that we are moving towards the back direction. And if you are moving in the back direction, then we need to integrate our traditional medicine into the modern medicine. And in this direction, several efforts are being taken. Here at All India Institute of Medical Sciences, we are developing one department, whole department of Ayush. And you will be aware that Department of Indian System of this Ayush was basically, there is a whole department of Ayush of the Government of India. And at AIIMS also we are developing Department of Ayush. And this Ayush was basically initially with the name Indian Systems of Medicine and Homeopathy. And this was renamed the Department of Ayush in 2003. And this Ayush basically stands for Ayurveda, Yoga and Naturopathy, Unani, Siddha and Homeopathy. And it is being set up in all new AIIMS. So in the same directions, there is a Center for Integrative Medicine and Research at AIIMS New Delhi. And what is the purpose of this Ayush? The purpose of Ayush at All India Institute of Medical Sciences is to understand the feasibility and applicability of making Ayush a cost-effective and mass healthcare modality, and to develop scientifically validated holistic medicine solutions for the disease states, and to produce Ayush-specific skilled human resources, and also to develop collaborations with the clinical and research departments in AIIMS to produce acceptable scientific evidence for traditional medicine system. And this purpose is also to position Ayush system of medicine as the preferred system of living and practice for attaining a healthy society. So here coming to some of the ongoing work in my laboratory. Basically, we are working on the breast cancer. We work on different aspects of the breast cancer. We try to understand the pathogenesis of the disease with respect to a class of a non-coding RNA that is called long non-coding RNA. We also try to develop some diagnostic and prognostic biomarker, basically long non-coding RNA-based biomarker. And finally, we also try to develop therapeutic aspect, and where we basically focus on the natural products. And because the focus of this conference is about the nutraceuticals, I am going to talk about the third aspect, that is therapeutic aspect. So if you ask a question, why breast cancer? Because if you see the breast cancer, this is one of the top cancers, both in terms of cases and the death that has been reported. And so that is the initially cervical cancer was the first top cancer, but now the breast cancer is the top. And here are some of the popular victims of the breast cancer. You see here is the model Susan Komen. You will be aware that she was diagnosed of breast cancer at the age when she was only 30 years old. And 3 years later, she died of the cancer. And to fulfill her desire, her sister, Nancy Bankland, is running one of the largest breast cancer foundation, just as Susan Komen Foundation in USA. And these are other popular victims. What I'm trying to tell here is that breast cancer, like other cancer type, does not discriminate between rich people and poor people. It can attack any kind of people. And if you see the predictions of breast cancer, per 100,000 in 35 years in different countries. In the past years, what has happened? So in developed countries like USA and Canada, there is decline in the breast cancer. But if you see the developing countries like Thailand and India, there is increase in the breast cancer incidence as well as mortality. And if you see the predictions of breast cancer mortality in the developed versus less developed countries, during the next few years, it is projected that it is going to decrease by 30% by 2030 in the more developed countries. On the other hand, in less developed countries, it is projected to be increased by more than 70% by 2030. And by 2030, 85% of all cancer deaths may be occurring in low middle income countries. And the reason he is here, this is because of the delay in accurate diagnosis. And this is because of lack of awareness. And as we adopt Western lifestyle, that is consumption of saturated fat, calorie dense food, and lack of physical inactivity, physical activity. These are some of the regions. So as you can see, considering all this, there is an urgent need to develop some novel molecule. And here I have already shown the potential of the inhibitors, potential of the molecules derived from the mother nature, that is natural products. And in our laboratory, we basically work on the one aspect of the basically chronic inflammation. And here we basically target to one pro-inflammatory transcription factor that is called NF-kappa B. And the reason is this is because these agents are multi-targeted, and we all know that, as I explained, that breast cancer is a disease that is caused by dysregulation of not only one or two genes, it is caused by dysregulation of multiple genes. Therefore, we need something that can target multiple pathways. These are relatively well safe. Their safety has been well tested because this has been used over the years. These are inexpensive and these are relatively readily available. And according to one report, more than 70% of the drugs that are derived, that are currently in the market, they have been derived from the nature. So here I already showed in my yesterday that panel discussion about the landmark of the nutraceutical research. If you see, so this is here one thing I would like to mention, that during COVID-19 pandemic period, the demand of nutraceuticals has basically increased more because of their use in immunity cancer as an enhancer of the immunity. And here is again, I would like to focus, give emphasis about the importance of the nutraceuticals, about the natural products. We will be aware that ivermectin is a compound that is basically derived from the mother nature, and this is again used for the this is basically used for the treatment of reduce the blindness and lymphatic fibrosis as well as several other parasitic diseases. And here are our own book about uses of natural products. We edited 2 books with Dr. Agrawal about the importance of the nutraceuticals and agents derived from the mother nature, that is anti-inflammatory nutraceuticals and chronic diseases, how we can use different nutraceuticals to cope up with the inflammatory conditions, and also the drug discovery from mother nature. So here in my presentation, I'm going to talk about a molecule that is some of you will be aware, and this is called basically piperlongumine. It is basically piperalkaloid, and what we did here is that, you will be aware that doxorubicin is a molecule, is a drug that is used for the breast cancer. However, what happens that over the period of time, the patients develop resistance to the doxorubicin. So our focus was that by taking piperlongumine, can we enhance the efficacy of doxorubicin, or can we cope up with the resistance situation? And so this work we did in breast cancer, and you will be aware that this piperlongumine is basically derived from a plant that is called Piper longum. And this piperlongumine is another reason is that, I come from a place that is called Assam as of now. So this plant is basically very common in Assam and Meghalaya, although it is also present in other states like West Bengal and UP also, but this is very common in Assam. So this is basically, this piperlongumine is derived from the roots of Ayurvedic plant that is called Piper longum. And if you see the discovery, this molecule was discovered way back in 1961. So the biological activity of this molecule was reported only during the last decade. The first breakthrough paper that came was 2012, and that was basically one PNAS paper. And then this activity of piperlongumine against breast cancer and the underlying mechanism is not thoroughly investigated, and this made us to work on this. Another thing is that whether this PL can enhance the sensitivity of breast cancer cells to the doxorubicin is also poorly known. So this was another reason we worked on this. So here I'm going to show some of the data only because I thought I have only 15 minutes, so I will not show much data. So here is one work we did basically with the animal, and the animal we basically used was the ESC model. What we did, we took the ESC cells and we injected into the mice, and these after the And after the 5 days, we randomized the mice into these 6 different groups. One group was control. Second group was PL, where we administered to the mice piperlongumine at 10 mg per kg body weight. Then we took another group where we administered doxorubicin at 1 mg per kg body weight. And then another group was piperlongumine at 20 mg per kg body weight. And then another group was doxorubicin at 2.5 mg per kg body weight. And finally, there was a one combination group where we administered to the mice piperlongumine 10 mg per kg body weight, followed by doxorubicin 1 mg per kg body weight. And this was basically alternate days. Means 3 days was piperlongumine and 2 days was doxorubicin. And over the period of time, over the period of 21 days, we monitored the mice for tumor development and also their behavior. And after 21 days, we stopped the treatment, and then after 23 days, we examined several endpoints. At the end of the experiment, as you can see here you see the control group where only the vehicle was administered to the mice. This is the tumor size. And this is basically the piperlongumine 10 mg per kg body weight. You can see it is a little effective. And piperlongumine 20 mg per kg body weight, there is effect. And doxorubicin, you can see at 1 mg per kg body weight, there was some effect. However, at 2.5 mg per kg body weight, there was no effect. And when we took the combination of two molecules, that is PL10 and doxo1, then there was almost similar effect as we got with doxorubicin 2.5. So this is one observation we got. And then over the period of time when we measured the growth of the tumor, as you can see here, this is the control group, this one. And these two are basically the individual agent, the lower concentration. And at the higher concentration of the individual agent, this is the growth of the tumor. And this is the last one where you can see the combination, the tumor growth was suppressed. This is at the last day of the experiment, the tumor weight in terms of gram. And you can see this is your control group. These are the two piperlongumine 10 mg per kg body weight, doxorubicin 1 mg per kg body weight, piperlongumine 20. So what you can see here is that piperlongumine combination group, you can see that tumor weight was significantly, drastically reduced as compared to the individual agent. The next parameter we examined was about what we found was that this piperlongumine was found to improve the body weight and food intake of the tumor bearing mice. As you can see here is the body weight. When you give your piperlongumine, there is increase in the body weight of the mice, and this was basically significant. So, and another thing is that food intake of the mice was also improved by giving this molecule. So, as I said that with cancer, there are several associated symptoms like body weight is reduced, the food intake is reduced. So, these are the, at least in the mice, we see this, that piperlongumine can improve the body weight as well as the food intake by the mice. This is, we also examined about metabolic parameters, some metabolic parameters in tumor bearing mice, and this basically we examined cholesterol level, we examined this triglyceride level, and also we examined that glucose level. And as you can see in the serum, basically here the triglyceride level is, cholesterol level is down, and then this triglyceride level is down, but glucose level is little high. At the cellular level, we examined several parameters, and one parameter we examined was about if PL can suppress the NF-kappa B activation that is basically induced by TNF-alpha. You are already aware that TNF-alpha is one of the well-known inducer of the NF-kappa B. And here, as you can see in that TNF-alpha alone group, what is happening that NF-kappa B, that is P65 subunit, is translocating into the nucleus if we expose the cells to the TNF-alpha. And here, piperlongumine alone is not doing anything. However, if you pre-treat the cells with piperlongumine followed by TNF-alpha, the translocation of P65 that was happening from cytoplasm to the nucleus, that was basically suppressed if the cells were pre-treated with the piperlongumine. So this is one parameter, and we also found that by doing molecular docking studies, we found that this piperlongumine can physically interact with two subunits of NF-kappa B, that is P65 and P50. So, and we also did several other computational analysis, like we examined whether this piperlongumine can physically interact with other molecules that are related with the NF-kappa B signaling pathway, like I-kappa B alpha, IKK alpha, P65, P50, TAC1, and also the GLUT1, that is basically a protein that is involved. That is basically a channel protein that is involved in the transport of glucose inside the cells. And we found that it does interact with these molecules. So another aspect that we have been working in our laboratory is about long non-coding RNAs. You will be aware that basically there are two types of non-coding RNA. One is called basically microRNA, that is well explored, and long non-coding RNA is another class of non-coding RNA that is relatively new field. And so here we are basically this long non-coding RNA we are using in other aspects also, like trying to understand some aspect of the disease and also to develop some markers with clinicians. So you will be here is basically, if you see, there are certain long non-coding RNAs that are associated with NF-kappa B signaling, like MALAT1, and this is one project is running in my laboratory. So if you see the NF-kappa B associated link RNAs, they are associated not only in cancer, these are also involved in several other diseases like cardiac diseases, diabetes, aging, PCOS, and several other diseases. And this NF-kappa B basically, NF-kappa B and long non-coding RNAs, they basically crossed up. So, and these are, when you see the long non-coding RNAs, where they are having both oncogenic and tumor suppressor function. Some long non-coding RNAs are having both oncogenic and tumor suppressor functions, as you can see here. And so here are some of our articles that we have written about the potential of long non-coding RNAs in the cancer patients, and also how we can target long non-coding RNAs by phytochemicals for cancer and other chronic diseases. Here is one of our recent book about clinical applications of non-coding RNAs. And so here you can see, so what we have developed in our laboratory, we have developed a platform where we can examine the expression pattern of 200 long non-coding RNAs, and this is a PCR-based array. And using the same setup, what we did, we exposed the MDMB231 cells to our molecule, that is piperlongumine. And then after 24 hours, we examined the expression pattern of these long non-coding RNAs. And what we found was that one long non-coding RNA that is called Gas5, this long non-coding RNA was basically upregulated. MEG3, another long non-coding RNA, was upregulated. And on the other hand, another oncogenic long non-coding RNA, that is H19, was suppressed. So we further explored this aspect about that. We use some siRNA experiments also, where we have some silenced Gas5, and then we have examined the functional assays like proliferation and viability assays, and we found that Gas5 is really involved in that this piperlongumine mediated anti-cancer activities. And here another aspect we examined about that acidosis. You will be aware that acidosis is the aspect that is basically involved in the tumor invasion and metastasis. And you can see that here what we found was that if you expose the cells under piperlongumine, that extracellular glucose was basically increased. Okay, and at the same time that extracellular pH of the extracellular pH is basically of the cells is basically acidic, and that is basically because cancer cells basically heavily depend on the glucose, that is Warburg effect, and during the process they produce several lactic acid, which is basically exported outside the cells, and that is again through the channel that is called, yes, okay sir, I'm going to finish. So, and so here basically what I am trying to explain is that here you can see that extracellular pH was basically from acidic to alkaline conditions we found, and we found that GLUT1 and MCT4, that are basically the channels that are involved in this process, that were basically the expression of this channel was reduced by piperlongumine. So, and also for tumorigenic proteins, we found that this can modulate the several tumorigenic proteins, and the data is here. So I think I will move on this with this data considering the time. And also we found that this piperlongumine using computational analysis, this possesses drug-like properties. So to cut the long story short, based on this data, what I have shown and what we have found that piperlongumine is basically a multi-targeted molecule. And this can basically does several things. It can basically inhibit NF-kappa signaling that will basically lead to the suppression of tumorigenesis, process of tumorigenesis. It can modulate long non-coding RNAs, and it can also induce apoptosis. And in animal models, these are producing several effects, as you can see here. So this is again some other molecules we are working, and I have been working on agents derived from the neem, and we have found that one molecule, TPA, is more effective. Yes, sir. So here I would just move on. So with this, I would like to acknowledge my group. These are my students. Some of these, the first 4 students are already completed their PhD. 3 are working. 2 are here. And I am been lucky to be surrounded by good people, both in India and abroad. And funding-wise, my laboratory is supported by these funding agencies like SERB, Indian Council of Medical Research, University Grant Commission, and Indian Council of Medical Research and Institute of Eminence. Thank you so much for your patience.
If you have any questions.
Yes, sir.
Dr. Gupta, very promising, but I heard you at caveat, I know nothing of this piperlongumine, whatever it is. To me, it appeared to have a more global effect rather than a specific. And this conclusion, and that's what at the end, I don't think you left anything which does not affect. So because you measured the NF-kappa signaling particles, and I would say provocative that because you knew that this was point cancer. But if you have checked other signaling pathway, I'm sure you would have found effect there as well. So I mean, what is your proposition?
My proposition, as I said, are multi-targeting. Multi-targeting is something that is required. And you are very right that it is not doing only NF-kappa B, because our focus is NF-kappa B nature. So we need something that is basically multi-targeting.
I'm saying that the audience must go with the impression that here is a herb which has a multi-potent capability. So they shouldn't go with the impression that this can only be used for breast cancer.
No, I am not claiming that this.
No, you are not claiming. As an audience,
there are several other work that has to be done. But based on only cell line and animal work, we cannot claim that this is a potent molecule. There are several other toxicity associated work. And if you see, sir, piperlongumine is really a promising molecule, and some clinical benefits also.
So that is one question, if you permit me. What is the survival go-up treatment? Yes.
So basically,
Sorry, yeah.
so this is basically working for GLUT1, sir. GLUT1.
Good for.
GLUT1.
Okay.
GLUT1. And also this has effect on the MCT4. So a GLUT1 is basically the one that is involved in the transport of the glucose from outside inside the cell. And MCT4 is basically involved in the transport of the lactic acid from inside the cell to the outside.
Thank you. It was a nice presentation, Dr. Gupta. Our next speaker.
I would request Professor Mazumdar to facilitate Professor Gupta for giving such a wonderful talk.
Sure. Thank you so much. I am honored.
Next speaker is Dr. Rupesh Srivastava. He is from the Department of Biology.
From the Department of Biology Sciences.
Sure, sir.
15 plus 5.
Yeah. No, so you do. Yeah. Permitted, yes. So very good afternoon to all of you. So am I audible? Hopefully. Yeah. So I thank the organizers, especially Dr. Professor Alok for giving me this opportunity, inviting me here. So without wasting any further time, I would like to start my talk, which is basically on Bifidobacterium longum, how it modulates your gut immune axis, specifically your regulatory B cells, Treg and A17 cell axis, and inhibits inflammatory bone loss in osteoporosis, and the mechanism of its action by ILA. So before going, so I'm taking you off some, basically we have been talking about cancer, but this is something which is very less talked about in all of the international conferences or the domestic conferences also. So this is why I'm basically working on bone health. So this is very important to all of you because it's going to be 1 in 5 individuals, specifically men, and 1 in 3 will be affected with osteoporosis related fractures in the lifetime. So this is a huge economic burden. So why I'm talking about this is because our skeleton is only found once we are born and it supports us throughout our lives, and we only have it one, regardless of our age, nationality, gender, race, and anything which we can take granted for. So that's why it's very important. And now coming to the bone health, why are we working on osteoporosis? So because it is a silent killer, we will never know it unless and until you will have a fracture. So it's chronic bone disease, and if you see the prevalence of osteoporosis, you can see that more than 200 million people are already suffering from osteoporosis, and this is a data from other countries where we are not listed there because, and we are the most populous country in the world now. So the data will be, I think, a double or triple of this because every 7th individual on this world is an Indian, and we have been taken the data from the Indian perspective because we don't have the data. So there is almost 1 fracture occurring every 3 seconds. And if you see that in India also, because we have a lot of people who are going towards aging and the population is going up, and we have almost 25 to 60% of the Indian postmenopausal women which are going to be affected with osteoporosis related fractures. And this is very important data. If you see here, hospitalization. So all of you must be thinking that heart disease or maybe stroke or maybe cancer is the one which is basically having most number of hospitalization, but this is not the case. If you see, the most fractures in hospitals are due to osteoporotic related fractures. And this is huge. If you see 43% of patients are admitted in hospitals due to osteoporotic related fractures and not due to your cardiac malfunctioning or your stroke or your breast cancer. I think, I'm sorry for that because some of the visibility is not that much clear, but that light sea green color is basically your breast cancer, the cancer which is most common among the women. So this is the fact according to International Osteoporotic Foundation. The data is as true of 2022. So we have 1 fracture going to occur in 3 seconds, and 1 in 5 months and 1 in 3 women in this auditorium will be affected by this. So be ready for that. So huge economic burden. And if you see 130 USD just due to your fractures. So my lab is in Department of Biotechnology, AIIMS, New Delhi. So it is translation immunology, osteoimmunology, and immunoporosis lab, that is TIOL lab. So we basically coined this term immunoporosis. Recently in the last 4 years, 2018, we coined this term, we proposed this term, this concept, and we are taking it to greater heights now. If you can see here, immunoporosis is basically what we call as immunology of osteoporosis. Okay, so earlier, very few people, or maybe none of them, or maybe most of you must be unaware that osteoporosis is also having an immune component also involved. Because you will think that it is only loss of estrogen that is basically leading to osteoporosis or bone fractures, but this is not the case. Immune system has a strong presence in the pathophysiology of osteoporosis, and this led me to coin this term that is immunoporosis. Both your adaptive immune system and an innate immune system are very much involved in an inflammatory bone loss in case of osteoporosis, and we have a number of publications. We have already proved that, and the world has also come to know that immunoporosis is a field, because if you type immunoporosis on PubMed, you will find 10 publications with the name they have taken up after we coined this and proposed this. So this is already well accepted now. Apart from this, we are working on every aspect of bone biology. If you see here, osteomicrobiology, toxicology, infectious diseases, how they are affecting your bone health. And also we are into how we can basically develop certain nutraceuticals or therapeutics from the natural sources, or nutritionally, or certain other compounds which can basically inhibit osteoporosis, bone loss and osteoporosis. So now, we have already established this Treg and Th17 cell axis. This was the start how immune system was proven by us that they are having a very strong presence in case of osteoporotic inflammatory bone loss. So we all know that these Treg and Th17 cell balance is important in most of the inflammatory conditions or diseases. But if you see here, this is what happens in case of inflammation. The balance is gone, and that has basically led to a lot of inflammatory diseases. And this is true in case of osteoporosis also. And we have in the last 5 years made this again and again that this is a very important axis, and this can be taken and this must be taken care of if you really want to inhibit bone loss in case of your osteoporosis. So when we were discovering all these things, Treg and Th17, basically being an immunologist at heart and also well trained in bone biology, I thought it cannot be only T cells which are very much important for your regulating such an important inflammatory axis in osteoporosis. There could be other cells also which needs to be discovered, and we are having that burden or basically that responsibility to discover the role of each and every immune cells because we started the field. So then what could have been the next cell in my mind, and that was basically it could be none other than your B cells, because T cells are adaptive cells. And B cells are also other adaptive cells which are a better hierarchy mein thoda upar hote hain because they can regulate the differentiation of both your regulated T cells and Th17 cells. And these were discovered in 2002 and they were named as regulated B cell in 2002, and after that subtype of Bregs were discovered in 2008, that is your B10 Bregs. They specifically secrete IL-10 for most of their immunomodulatory functions. So we know that starting from allergy, asthma, your graft rejections, and your almost all of the autoimmune diseases, tumors, and everything, regulatory B cells have a very strong, proven role there. But in the case of osteoporosis, it is not known because nobody have ever thought about this. But basically, osteoporosis could be also an immune-driven disease. So this was the question which we asked, and we started from there. So the first question is, do Tregs have any role in osteoporosis? For that, when we have to go to the in vitro cultures, first we have to generate Bregs, authentic Bregs. So what we did, we took splenocytes, we sorted the B cells, we stimulated them with IPS for 24 hours or 48 hours, and then we checked for the presence of IL-10 among these B cells, and we did found that this was highly significantly enhanced, and the IL-10 secretion was significantly enhanced in these B cells. Moving ahead, the next question was, do these Bregs have any osteoclastogenic, anti-osteoclastogenic activity? Because osteoclasts are the cells which are basically eating up your bone, responsible for your bone loss. And another cells, osteoclasts, are basically forming bones. So these two are the cells which are basically at the core of any preventive strategy for osteoclastogenesis or osteoporosis. So we checked, first of all, whether these Bregs which we have generated in vitro are having any anti-osteoclastogenic potential or not. So what we did, we co-cultured these Bregs generated and the bone marrow from the bone marrow monocytes, you can say from the mice, and we co-cultured them at different ratios, and we were really excited to. So if you can see the red color cells, these are your osteoclasts, and they have been stained with TRAP, that is an enzyme present in the osteoclast, and this is a very much strong, the only marker for your osteoclastogenesis or the osteoclast TRAP. These are multinucleated and they can be more than 200 nuclei in a single cell. They're syncytial, they are multinucleated, they fuse, all the osteoclasts they fuse and they are huge, and then they resorb the bone. So you can see here in the presence of this positive control, a lot of TRAP positive cells are there, but the moment we are co-culturing them with in a ratio dependent manner with the Bregs, this is basically inhibited. And this was again confirmed with the F-acting ring, because F-acting ring is an important functional marker for these osteoclasts, because this is only due to the formation of this F-acting ring that they can absorb the bone efficiently. So this was a very eureka moment for us, because this was a contact dependent manner. Then we thought maybe it could also be in contact independent manner. For that, we did the same experiment in Transwell setup. And we did find that in the contact independent manner also it was inhibiting, the Bregs were inhibiting osteoclastogenesis. Then the next question was, since these Bregs are known to have their functions via either IL-10 or 35 or TJ beta. These are only 3 molecules which are known to have, by which the BRAFs can have their immunomodulatory effects. So first we checked for IL-10 because these were the main, the hit is 80% of the functions are due to IL-10. So for that, we did with an anti-IL-10 experiments, anti-IL-10 antibody experiments, and we did find that the moment we are adding anti-IL-10 antibody, all these inhibitory effect is abrogated if you see with the respect to isotype. So this was again confirmed, okay, IL-10 is a molecule which is basically having the anti-osteoclastogenic activity via these BRAFs which have been formed. So our calculations are that they can emit more than 30 osteoclasts. So that is a huge number, 30 times they are potent enough. So now again moving ahead, so what is the case in vivo? So we developed the postmenopausal osteoporotic mice model by removing both the ovaries, and we authenticated that with the help of your estrogen levels, and also did SEM and AFM, all those things, and also the gold standard is microCT. If you can see here, these are all the control, and these are all the LP5 vertebrae, which is the gold standard, which has been needs to be assessed. You can see a large number of tubercules or the bone deformities are there. Also in the femur tuberculi, tibia tuberculi, corticals also, both the femur and tibia. So this, after confirming this, after developing this model, we look what is happening to the status of these beaten BRAFs in your osteoporotic mice and the control mice. And if you see here, we did both of these BRAFs, the total B cells which are secreting IL-10. You can see in our atomic conditions that is significantly going down with respect to both in your bone marrow, which is the primary site of osteoclastogenesis and bone loss, and also your spleen where the induction of BRAFs is basically taking place. So both of these places were having a significant reduction in your BRAFs number. And the other B10 population, which is basically characterized by your CD19, CD5 positive, 1D high BRAFs, they were also having a reduction in the population of these BRAFs in both these populations. The next question was, so estrogen is basically, we are taking out the estrogen due to the removing the ovaries. This same effect is also possible when we are doing in an in vitro ex vivo cultures, and we did find that when we are culturing these BRAFs in the presence of estrogen, all this inhibitory effect is going down, and the BRAFs are able to proliferate or differentiate in that manner. And this is very much, if you see, is correlated with these values are correlating with in vivo and ex vivo both conditions. So now, the next question is, because in case of rheumatoid arthritis, what happens that both the numbers of BRAFs and both the functions of BRAFs, they both are affected. So the number is already going down. The next task was whether the functions are also compromised or only the numbers are affected. So what we did was that we picked both the sham and the mice, and we isolated the spleen, and we took a control mice, and we did take the spleen and we co-cultured that. We isolated BRAFs from here, both of these conditions, and co-cultured them with the osteoclasts from these normal mice, so that we can see whether the BRAFs from the osteoporotic mice and from the SAM mice is basically going to inhibit them. Now, coming here, so BRAFs are functionally compromised. If you see here, they are not able to inhibit the proliferation of T cells in case of your overectomy mice, 65%. And 53 is normal control mice, control sham cells, they are able to inhibit that. And apart from this, the differentiation of the Tregs and TS17 cells were also compromised. The BRAFs immunomodulatory activity, the obese BRAFs were unable to induce induction of Tregs as was expected. In fact, they were basically enhancing TS17 cell population because they were unable to control your Treg cells. Now coming in, so there exists a BRAF Treg TS17 cell axis in osteoporosis, and one BRAF can inhibit 30 osteoclasts, and one Treg is already known to inhibit 100 osteoclasts from other studies and from our own also. So now the data is BRAFs can induce Tregs threefolds. So ultimately, one BRAF can inhibit 330 osteoclasts if they're also affecting your Tregs. So that's a huge potential of these. So now the model is something like this. The BRAFs are also there and helping the Tregs to combat the osteoclastogenic activity of the TS17 cells. Now moving ahead, so what is happening? So if this, so there exists so this is an immunoporotic axis which we called it, that BRAFs can basically regulate osteoclastogenesis, and all the known drugs, bisphosphonates, teriparatide, democenazole, bisphosphonate, they all are basically somehow they are affecting this immunoporotic axis, but we don't know them. So if we can tweak this axis, then basically we would be able to do an anti-osteoclastogenic effect or anti-osteoporotic medications can be formed. So how can we do? But because we are already having these drugs, but they have a lot of side effects. We can have a lot of side effects, including depression and heart attack also, and breast cancers also. So can probiotics be the game changer, because we are working on probiotics from the last 7 to 8 years. So now coming, so we took Bifidobacterium longum as a probiotic of our choice because of its it's very well studied. So what we did all these experiments again after treating these BRAFs with BL, inducing these BRAFs in the presence of BL. So what happened if you see, if you're inducing this BRAF, these have a strong anti-osteoclastogenic activity with respect to your only BRAFs. So BRAF sector is driven are not inhibiting osteoclasto that much great, but if you are treating these BRAFs with BL, the potential, the immunomodulating potential of these BRAFs is enhanced significantly. So again, these enhanced immunomodulating potential of BRAFs is also affected in case of your Tregs and TS17 cells. If you see here, both your Treg and TS17 cells are strongly modulated by this BRAF population, which have been primed with your bifidobacterium long-term supernatrants of these cultures. So again, what we again did the same experiments with a BL group, OX plus BL, and we did find that BL was inhibiting osteoclastogenesis in case of osteoporotic mice model, and this is how this is effective. And this is how BL is modulating Treg and TS17 cell axis in mice via regulating your Tregs and TS17, as we already know, and this was in a much higher significant manner if you're treating that with BL. So now, BL basically have enhanced BRAF population also. They were enhancing, apart from Treg and TS17, they are also enhancing your Tregs population, if you see, sorry, BRAF population also here significantly. And this whole axis now become, apart from that, if you see, IL-17 and IL-10, basically IL-10 secreted by both your BRAFs and Tregs, and IL-17 is being secreted by TS17 cells. These were also significantly modulated. So it's just the beginning, and we published this paper just 4 months back in Frontiers Immunology. And also this, we got a best paper award for this for my PhD student Leena Sapra. She is sitting here, and this is one of the most prestigious awards given to the AIMS PhD students for their best papers published. So this is just 7 days only. So now coming next, what is the active metabolite for these BL conditioned media? So what we did find was that BL was secreting ILA when we checked with the help of mass spec. The ILA, the indolactic acid concentrations were very much high in these culture supernatants. And we did all these experiments in vitro again with ILA. Interestingly, or heartbreaking, ILA was not inhibiting osteoclastogenesis in a dose-dependent manner. So what is the role and how they are affecting them? The question is, so they are not going through osteoclastogenesis. They don't have a very strong anti-osteoclastogenic activity, but they have a strong induction of B regulatory cells. And if you see here, they are able to induce significantly enhanced regulatory B cells population, and these BRAFs which have been primed with ILA are having a further enhanced anti-osteoclastogenic activity. If you see here, we're treating these BRAFs with ILA, they have a strong ILA, strong anti-osteoclastogenic activity. So ILA, why BRAFs are basically affecting these conditions in case of your immunoporotic mice, and this again we did that. We put in the ILA and we did find that 4 times your BRAF induction that is one in the bone marrow, the blood, and the colon, and this is coming now. So also the gut integrity was enhanced after giving the ILA treatment because that is much lost in case of osteoporosis. We did a fixed dextran labeling method in the mice. So the take home message is, if you are giving your tryptophan-rich diet, for example, with the case of eggs and other things and oats, and then BL will treat them and ILA will be formed, they will be enhancing your gut integrity and maintaining gut integrity. Apart from that, they will be also immunomodulating of BRAFs. BRAFs will then have a strong induction of Tregs and inhibition of TS17 along with your inhibiting osteoclastogenesis also. So this is how this is basically. So now we again check that in case of your osteoporotic patients, and yes, the BRAF populations are compromised in case of your humans also. From 6, it is coming to 2.28. And recently we got a clinical trial approved from ICMR for studying this axis in case of your osteoporosis. And we are going to start them. This is the contributions we have already been featured on the cover page of Immunity 2019 in the 25th issue for a contribution as immunoporosis, the field which you call and other things. And we have been credited with immunoporosis apart from gut immune axis, Treg TS17, BRAFs and Treg TS17 cell axis, along with employing probiotics and therapeutics for osteoporosis, in which we pioneered. And these are some of selected publications, and I thank all of my students. This is many young PhD students have also joined in, and I thank all of my funding agencies. I have funding from almost all the agencies, Ayush, ICMR, DBT, DST, AIMS, UGC. And so love your boss because they are going to protect your future. Nobody else is going to come, and all of you are going to be affected by this. So be ready for that. Thank you.
Dr. Srivastava, one quick question. Yeah. So if you include the B cells with APS, they will develop into BRAFs, and these B cells are also able to produce antibodies?
So this BREC, the method of induction of BREC is a T cell independent method.
Yeah, because we use APS to get T cell element.
They used to produce antibodies, but those antibodies are not having a high affinity or affinity in all those things are not as high as with respect if you're activating them with the T cell dependent manner. For example, anti-CD40L and all those things.
That's obvious. The other question is, what is the phenotype of these BREC cells?
So these BRECs are CD19 positive, CD5 positive, CD1D high, and IL-10 positive. So these are called as E10 BRECs.
Okay.
Specifically, if you activate them with LPs.
IL-10 appears to play a major role in this. IL-10 appears to play a major role in this. So what happens if you have an IL-10 knockout mice?
So if you have an IL-10 knockout mice, you will see, because people have already done that in IL-10 knockout mice, there is increased exhibition of these bone loss in those cases. In case of rheumatoid arthritis, people have done that because nobody have ever thought of osteoporosis as an immune mediated disease. So nobody has done this. But in case of RA, people have done that. They did find that there was increased bone loss in case of your IL-10 knockout.
Thank you, sir.
Yeah, thank you.
So,
I now request Professor
To felicitate Dr. Rupesh Srivastava for his outstanding talk.
okay. So today's last speaker is Dr. Soumya K. Patel, and I would request him to come and talk over here. And be bit careful about the timings because we have a valedictory session.
Yes.
Yeah.
Okay. So first of all, I would like to thank organizers, Delhi University, for giving me this opportunity. So my topic is exploring the meronome of Erica papaya with potential cross-kingdom human gene targets. So now as we live in the era of the like genome, proteome, transcriptome, methylome. So I touched one of the aspects of the total heteroRNA, that is the meronome of the known plant, that is the Erica papaya. It is a papaya that we eat, and whether it is having any first kingdom role in the human genes. So first of all, the study center around hypothesis that we are what we eat. So ultimately, Ayurveda and the other Hindu remedies work on the people's nature more. So ultimately, we are what we eat, and the question is, do plant-derived microRNAs have any role in regulation of the human gene expression in a kingdom and what? So to address this question, the study was undertaken using the Erica papaya as a model plant. So as we all know, the Erica papaya plant, it falls under the kingdom, and India is a major source for the production of the Erica papaya. And in terms of the nutraceutical value, it has all the nutrients like magnesium, iron, calcium, and other vitamins. And we talk about the phytochemicals. Also, it is having a rich in phytochemicals that have a certain disease ailment. So, I think all the parts of this papaya that are being used for the treatment of the certain diseases has a medicinal impact. So what we are interested in taking the picoRNAs from this plant. As we all know, these picoRNAs are non-coding RNAs and are between 18 to 22 proteins. And we aim this means for the potent as an potent regulator of the gene expression in human. So basically, this is the biogenesis pathway of plant and animal picoRNAs. And while we have taken these studies, right? So this area is very new. Globally, very few labs are working on this area. It was started in 2012 when first discovery of the mRNA 168 from rice in mammals that is proven to regulate the certain human genes, right? And so on. And we started this work in 2017, and we could publish our first paper in 2019 that maximum bacillus, that is the tulsi that we eat, that found to have a multiple picoRNAs that can regulate the certain human genes. So in the presentation, I have focused on two objectives that we did. First of all, one of it is that is the computationally we could identify certain novel picoRNAs from through the EST as a reference data. They express sequence tech data, and the second is the sequencing total small RNA sequence of the plant, and to study the conserved and the novel picoRNAs. So while in the first study, we have taken the model kingdom as a 2D planting and the EST data set of Erica papaya. So from that, through the homologic searching and the several bioinformatics studies, we could identify some 9 novel picoRNAs from this prediction. As you can see here, we have set the certain parameters to fetch this kind of picoRNAs as the minimum length, and the minimal folding energy and the adjusted minimal folding energy. So these are the two key picoRNAs, the CPA MIR 36290B and CPA MIR 6034. So these two key picoRNAs from the all the available EST data till now, they are regulating, they are targeting 37 human targeted genes. And along with that, also we did the gene ontology studies on the basis of the annotation of these target genes. That is in terms of the role of the genes in the cell component, molecular function, and the biological process. Along with that, also we did some protein-protein interaction studies and the network pathway analysis studies. So you can see here the protein-protein interaction of the Erica papaya derived targets emerge as a inhibitor genes. And these are the top hub nodes. So ultimately, from the studies, we can say that we found the two novel waves targeting 37 genes in human, and ultimately these 37 targeted genes are out of the 18 genes involved in the predominant cancer pathways and the platelet activation. Our second objective, that is total small RNA sequencing of this plant, and to study their genome. So for that, we have isolated the RNA content from the plant, and through HiSeq NGS platform, we could have the sequential data, and through the bioinformatics studies, after doing some filtration and the other QC check, we found several high quality reads out of that. And we did certain bioinformatics studies, and that we have done some reference mapping with the available data, and from that we have so we did the last time studies and the MRAP studies. So from that we found 17 hits from that, and when we have against the genome, we found 49 novel RNAs. So together these 9420 hits and the 1798 hits, after the second chapter prediction, we found total of 49 novel RNAs from this. These are the list of novel mirs that we found. And again, when I talk about the commutation part, we did actually for the geoanalysis, that is in terms of the cell component, molecular function, and the biological process. So this is the again gene network sequencing showing the association between the significantly related genes. So these are the list of top hub nodes calculated through the topological studies, and this is the hilo tree for Carica papaya, which is of novel mirs and the reported mirs. So from these studies, we could identify 4 novel mirs and their human gene targets that are being depicted by this circus plot. So ultimately, from the studies, we could generate the 30 million reads using the NGS technology, and we have identified 49 novel mirs, and 4 of them were found to target almost 70% genes out of total predicted human targets. And the cross-spectrum studies of this novel Carica papaya microRNAs will have the implication in disease management with the diabetes, cancer, mental and the restricted detection. So ultimately, from these studies, the key findings are, as I have depicted, we studied the list of mirs by PST approach. We defined 9 potential microRNAs, out of which 2 mirs are novel, and from EST approach, this mirs plays a major role in the continuous process, disease association, and predicting the downstream signaling cascade via predominant pathways of cancer, and may have a functional impact on a human disease. From the second objective, we could identify the target genes constituted a significant role in the various downstream signaling pathways as W entry, nano, MAPK-AKT, PRK, along with the pivot activation and revision pathways. So these are the target genes which were involved in the various signaling pathways and acted as oncogenes. And finally, we also studied the common mirs involved via EST approach, the genome-wide approach, small RNA sequencing approach, and from reported microRNAs deposited in the mir repository. One of our key findings that CPA MIR 390 is common in all of the 4 datasets, and there's a potential value in terms of the expression of the certain genes. So from these studies, we could publish 2 papers from this data, and now we are validating these target mirs. We are making the in culture, we are taking the mimics of this microRNAs, and we are relating to the gene expression studies in terms of the luciferase gene reporter assay and the other assays. So, thank you.
Thank you, Dr. Patel, for this nice presentation. Okay. We come to the end of this session, sir.
So, may I request Professor Kumar to please felicitate Professor Patel for his
I'd like to thank the audience for being with us as well as the presentation. We started with Srivastava, we talked about the role of neutrophils in cancer and how tuning the neutrophils could serve as cancer. From Universal Medical Sciences, Guwahati. He made us aware of the causes of breast cancer, and we talked about the modern of medicines. Disease. This is followed finally, Dr. Patel from the present between the plants and the human system. And with this, we come to a conclusion. I don't know. I want to hand over to Mamta.
I would like to thank everyone present here to patiently listen to all the lectures that have been taking place. And I would also like to thank the chairs of the session. And taking this opportunity, I would like to invite Professor Rajagopal Raman and Professor Rajakarjan Shakarat to please felicitate the chairs of this session.
एक अच्छे से ले लो, चारों को लेके।
चारों को नहीं, वो अलग-अलग लीजिए। Please.
Thank you.
सर ये दिन का रहेगा तो सर आप कुछ कहना।
हाँ, ले आएंगे, सिर्फ पढ़ा तो जाएगा नहीं। अरे आइए, इसमें उंगली रखते हैं सर।
Thank you, sir.
इसमें ममता नाम लिखा नहीं, मैं दे देता तुमको। क्या करेंगे?